Well, hello everyone. Welcome to our Safer World by Design webinar for today. And today I'm very happy to have Paul Jennings with us from the Free University of Amsterdam in Holland. We've had a lot of interactions with Paul over the last few years on different projects. And for the first time on our Safer World seminar, we're covering the area of nephrotoxicology and some of the related issues that we share in this community related to mechanistic toxicology. Paul's been a leader of that and is always coming up, I think, with new ideas and new innovation in this area. So very happy to welcome him, and we'll have a presentation from him approximately 25 minutes. You can enter your questions into the question box at any time, and we'll have about 20 minutes of discussion at the end. With that, over to you, Paul.
Thanks, Barry. Just writing down my target time of 3:30. Okay, so welcome everybody. Thank you for taking the time to listen to me. So I'm going to talk today, well, Barry actually asked me to talk today about nephrotoxicity and models for nephrotoxicity. It's not the only thing we do in my lab, and I'm going to just quickly tell you all the activities we do just very, very shortly. I have a pointer, but it doesn't seem to work for some reason. Maybe it'll work now. Oh, yeah, that works. So I am a professor of molecular and computational toxicology at the Vrije Universiteit Amsterdam, and this is a snapshot of our group. And in that picture, we actually have a lot of visiting academics from the IN3 project because we had an IN3 meeting at the time. So who are we? Well, that's our team, and we are specialized in developing both computational and human in vitro, non-animal-based strategies for the understanding of molecular mechanisms of chemical-induced toxicity, so we can better predict chemical safety in humans. And to be more specific, we combine our research on phase 1 and phase 2 metabolism, but also on transporters, so xenobiotic transporters. And we're also very interested in beginning to look at the influences of polymorphisms, because people are different from each other, and we want to incorporate that difference into our strategies. We look at the molecular mechanisms of, so the molecular stroke cellular mechanisms of chemical and also nanomaterial induced cellular perturbations. We are specialized in kidney and liver. My background is kidney, but when I joined DeVu 3 years ago, I joined basically a dedicated liver lab. So that's nice. We have those two high specialities. But we look at lots of different tissues as well, lung, and I collaborate with many people who look at other tissues like blood-brain barrier with Maxime Coulot in France. And then Dan Gierke in my lab, he is a specialist in computational methods, and he looks at site of metabolism prediction, including molecular docking and molecular dynamics. Our laboratory is specialized in many sub-branches of this, including analytical chemistry, cell culture, toxicity determination, omic analysis, and as I said, molecular docking and dynamics. And we grow a variety of cells, human primary cells, human cell lines, and we are now getting big time into induced human pluripotent stem cells. My duty is 50-50 research and teaching. And so I really like teaching, and I like interacting with students and explaining things. And what we teach currently is drug-induced stress and cellular responses, replacement, refinement, and reduction of animal experiments and biomedical research. I have an advanced course that's based around adverse outcome pathways, and we also teach the principles of ADMET, and all of that at a master's level. And then we also have some teaching, or quite a lot of teaching in the bachelor's level. And those students also join us for their internships in our laboratories. And yeah, it's fun. Okay, I don't know who is in the audience actually, but I think most people who are involved in toxicology know that that Paracelsus was crowned the godfather of toxicology, because he had this interesting philosophy. So Paracelsus was from Egg in Switzerland, and he studied in Basel, and he lived quite a large part of his life in Salzburg in Austria. And he said this: "Alle Dinge sind Gift und nichts ohne Gift, allein die Dosis macht, dass ein Ding kein Gift ist." And to paraphrase, this means everything is toxic, nothing is without toxicity. The dose alone makes the poison. And it's an important thing that we forget about, that toxicology is not about the determination of a toxic entity or chemical. It's about knowing the uses of it and the relevant dosing of it, and at what concentration that you might be exposed to would then be toxic. So it's really about this concentration finding, that what above a certain concentration is toxic and what below isn't, and then compare that to what your potential exposure would be. Where do toxins come from? Well, there's natural toxins, radiation, metals, UV light, for example. There's biological toxins, bacteria, plants, fungus, and animals. If you think of frogs and venom from snakes, and actually some of them are quite brutally nephrotoxic too. And then we have, where we spend most of our time, is in synthetic man-made chemicals. And that's industrial waste products, from the manufacturing industry, the cosmetic industry, and then, of course, drugs. And we also are interested, of course, in the metabolites of all of these things, and especially drugs. How do you get exposed to toxins? Well, you can inhale them through the lung. You can ingest them where the mouth and the esophagus is affected. You can be in contact with them through thermal exposure. You can ingest them, so then you have it enters through your stomach, and that's how we take drugs usually. And then you have the first pass metabolism. So if you ingest something, it will normally, not always, but normally go to the liver directly after the gut, and then the liver is an expert in processing chemicals, especially chemicals that are lipophilic. And then, of course, you can be injected, but you can be injected not just in a hospital. You can be injected by a bite, a spider, or a snake, or something like that. Now, if we talk about, I was supposed to be talking about nephrotoxicity, but let's open it up a little bit. So, and I'm only an expert in human toxicity, which is challenging enough. But we talk about different levels of toxicity. So there's individual, as an individual human. So you have individual mortality. So are you alive or are you dead? You have individual morbidity. So how healthy are you? Then as an individual, you also have progeny. So your fertility. And then only for women. Then if a woman is pregnant, that's a very complicated developmental process, and it's quite easy to disturb. So we have to be careful with chemicals that pregnant women might ingest voluntarily or by accident. And then we have the entire population of that, of course. Now, if you think about no longer individual, and you think about an organ level, so we think about things like, for systemic toxicology, we think of things like liver, kidney, lung, brain, heart, but all of the organs are actually important. And then you can think of it on a cellular level. So as the smallest biological entity that's capable of replicating itself is a cell. And there's about 200 different types of cells in your body. And depending on where it is in the body and what it's exposed to and how it has developed, it will react differently to those chemicals. You can think subcellularly as well, so at a molecular level. So cells are just a bunch of chemicals actually that evolved, and this is the 2D structure of a DNA molecule, and other chemicals can actually bind to that and disturb it, and DNA toxicity is pretty bad. Okay, let's move slightly now to the kidney, and the kidney is it's special in that it's different to the other organs, but all organs are special, right? So I just want to say why the kidney is special. So the kidney is approximately fist-sized. You know, you have two of them, and they're here around the back of your body. And the main job is to regulate water and electrolytes. So it controls blood volume. But it has lots of sub-roles in that, so it also regulates acid-base balance. So the kidney tries to keep your blood pH at 7.4. The kidney also plays a pivotal role in phosphate, vitamin D, and erythropoietin regulation. And so erythropoietin is, as you know, is required for producing red blood cells. So the more erythropoietin, the more red blood cells. The kidney also reabsorbs essential substrates, including water, glucose, ions, amino acids, and vitamins. And it eliminates water-soluble waste products and xenobiotic, and it eliminates excess substances too. So if you take too much salt, it simply leaves into your urine, for example. Some numbers are interesting, that the kidney sees 1.5 thousand liters a day of blood. So that's 1.5 tons a day the kidney sees. And it filters 180 liters per day. The functional unit of the kidney is a nephron, and it's depicted here in this is a drawing I drew for a Henry Stewart lecture. And you can see the different sections. And it's like a tube. One tube, and it comes into the glomerulus, which is a professional filtering system, and then the tube moves things in and out of it until finally you have your urine. You have 1.5 million nephrons in each kidney, approximately. The kidney doesn't have a really good regenerative capacity in that the cells themselves can repair, and they do, and divide, but if one nephron is lost, it's lost forever. So on the bullet point 3 there, you can see that I'm saying that there's no de novo nephrogenesis after birth. So I'm not saying that the cells don't repair, they do, but if the damage is so bad that the nephron is ruptured, that's the end of the day for the nephron. And instead of having 1.5 million, you have 1.5 million minus 1. And we lose nephrons all the way through life, and chemicals can accelerate that process. And on this graph here, you see on the right-hand side, on the bottom of the screen, you can see that chronic kidney disease is a really heavily correlating disease with age, like many other diseases. So the nature of chronic kidney disease, and it's the thing we're trying to avoid, so we don't want chemicals that make this initiated or make it worse, it terminates in end-stage renal disease. So when you have end-stage renal disease, your kidney is no longer functional enough to do all those things I just told you, and then you either need to be dialyzed 3 or 4 times a week, or you'll need a transplantation. And transplantation is really the best option. Dialysis, you don't live well with dialysis, but it's absolutely necessary to maintain life. It's a major societal and economic burden, and we think that 10% of people are affected worldwide, and it's increasing simply because people are getting older. And that's the next bullet point. And we think it's also linked to neurodegenerative diseases such as Alzheimer's, because if your kidneys are failing, then you have more and more toxins in your blood system. You border on acid-base disturbances. Your blood volume regulation is poor, and you can lose essential vitamins in the urine if the kidney isn't good. So, and all of these things can exacerbate neurodegenerative diseases. And then chemicals, drugs, and environmental pollutants can also initiate kidney disease, and that's the area where we're in. So we have to understand it to predict it, and yeah. Now, this is just a little silly cartoon I made, but your kidneys are pretty vital, healthy organs. They use a lot of energy because of the transport mechanisms they have. It's ATP dependent. So the proximal tubular, for example, uses a huge amount of oxygen, but it's no problem because it's really, really well perfused. So you can have acute renal problems, or very acute ones like renal inflammation. And if that happens, if you look after yourself, it can be fully reversible. But if this progresses and the acute disease accumulates, it will initiate chronic kidney disease, and chronic kidney disease is not reversible. And as I said, you end up in end-stage renal disease. But even in this slightly sick condition, where it is reversible, you get all of these clinical symptoms, and immediately. Polyuria, glycosuria, metabolic acidosis. You lose, you have too much uric acid in your urine, and you lose things. And this can cause growth retardation in children. It can cause also rickets in children, and some nasty things in adults too. And for this to happen, you don't even need any cell death. So you don't need any cytotoxicity. You just need a hypoxic event, or not enough oxygen, or something else that happens to the kidney, that the kidney simply doesn't do its job, but the cells are all alive. They're just not being able to do those transport functions. So the kidney, the nephron, this is a closer view of the nephron, and on the slide you see I've drawn a blood vessel. So the blood is white blood cells and red blood cells. And the endothelial cells, basically in the glomerulus, they prevent the cells entering the nephron, because that would be bad. You don't want to lose cells in the urine. And the podocytes then filter the metabolites and the chemicals and water through, and they let nearly most things in. You see up on the top right what's excluded, and the podocytes do a little bit more additional filtering, but they keep the whole barrier intact. And then the proximal tubule is a professional transport machine, and it will bring back nearly everything that comes through under normal conditions. But as I said, if you eat too much salt or whatever, those pathways are overloaded, and the salt simply goes into your urine. It's complex though, so the transport mechanisms are highly complex, and they're all highly dependent on sodium, potassium, ATPase. And that's the one that needs a lot of energy, and those cells are stuffed with mitochondria, which I'll explain a little later. So that's my picture of the mitochondria eating fatty acids and oxygen. So how do we investigate that in our lab? So we have 3 different ways of doing it. So with primary cells, so they're cells that come from, remember, I'm only talking about human cells. We don't grow animal cells. So we can get people who had a car accident, or usually we get our kidney cells from renal carcinomas, because they're usually overcautious and take out healthy tissue as well. But that's actually becoming problematic, because the surgeries are getting better, and they can be more exact where they cut. We have cell lines where you make a cell permanent. So you trick it, you break a pathway called usually P53, and then it kind of becomes a cancer in culture, and then they grow forever. And that's a nice tool, but they're usually much more disturbed in their biological functions than primary cells. And then we have stem cells, where you can teach an embryonic stem cell or an induced pluripotent stem cell to become a kidney cell. And we're learning nice tricks how to do that. And Anya Vilmas from my team is heading those activities, and they're very exciting, because it's really fascinating that you can take a stem cell and make it a kidney cell by just giving it some growth factors at the right time. One of the cell lines we've used a lot comes from Vienna, a company called Eversight, led by Johannes and Regina Grillari. And they're a nice little SME. We work a lot with them. And what they did, they were basically involved in aging research. And they learned that if you overexpress telomerase, which is an enzyme that keeps in check the telomere, which are tandem repeats at the end of the chromosome, and they shorten with age. And if you just activate that system and add a telomerase, it will make them grow forever in culture. And so they generated this nice cell, and when they sent it to me, I was amazed, because it looked exactly like primary cells, and they form tissues, and they have domes, which is a sign of transportation, and they have trans-electric resistance. They have the right type of hormonal activities. Now they're missing some things as well, but that's to be expected. The one good thing about them is that they're not cancerous. So we've tricked them in a way that's a bit less brutal than normal, and this means that they have a normal karyotype at this level. So we know they do harbor mutations, but they're not terribly bad mutations. And just comparing the left karyotype panel to the right karyotype panel, that's A549 cells. and their karyotype is completely messed up. The chromosomes are all swapped around. There's even some of them have 4 chromosomes, but you should only have 2. So it's a mess, the A549 cells. I'm not particularly fond of that model. I think some of you who know me might have heard me say it before. So these RP-33 cells are beautiful, really beautiful. So they're tricky to handle in culture, but that's normal because they're not cancerous. So you have to really look after them. And we're professionals in looking after these cells. We have them in the lab all the time. And so when you see them first, they look like this at day 2. So they don't really look like epithelial cells. They look like more like fibroblasts. And then as we give them more time to develop the tissue, they start getting really compacted, and we call this a cobblestone morphology. And then they start to form these domes. And the domes is a raised area where water is reabsorbed, and it lifts the cells from the plastic. And we see all of these processes are correlating with a cell cycle arrest. So as they get older from the seeding time, they become cell cycle arrested, and they differentiate, and they start developing new features. Now, unfortunately, in the literature, nearly everybody, apart from our lab and a few other labs that I've had discussions with, uses them in the subconfluent state, which is very unfortunate because they're less kidney-like there. So a message to any of you, if you're using them, use them in the confluent state. And you can see here also in my image, here we stain for ZO3, which is called tight junction 3. And it's a protein that's associated with the tight junction, but it's also a protein that goes into the nucleus and drives proliferation. So as the cells mature, you can see that ZO3 is no longer in the nucleus, and it's now associated with the tight junction. So it's a nice little example of how they change over time. So I only have 6 minutes left, but I don't want to go fast or anything, so I'm just going to see how far I get. So interpretation of in vitro data is more complicated than you think. I thought I had a slide about, yeah. So we want to move from using animal experiments for a few reasons. One of them is ethical, of course, but another reason is animals are not humans. And the precision we have now at mechanistic precision, it's truly amazing with all these omic technologies. So then it's a kind of a waste if we spend all our time understanding the physiology of a mouse or a rat at a cellular level. So we really need to move to humans. And that's why we also don't grow any rat cells. And I said, these are the tools we have available: cell lines, primary cells, and induced pluripotent stem cells. And induced pluripotent stem cells we can get from living donors, and we can select. We can pick anybody. So I made this example. I could take all the people in this meeting. I think there's about 20 people. If you gave me about €100,000 in 6 months, I could have all of your cells in my lab, and I could make kidneys and livers and brain. Not all myself, but my colleagues would help me out. And that's a nice new thing, because normally for embryonic stem cells, they were from aborted fetuses, so the person never lived. And if we can correlate that with actually living people, we can see what pathologies they might or might not have, and then correlate that in a dish. So that's a very exciting branch of research. Now, why is in vitro more complicated than animals? Well, see, animals, we have context, right? So we know how much we give them, we know the exposure, it's just more normal how we do those experiments. And also, if we do, say, an LD50 test where half the animals are dead, it's precise. We know that half the animals are dead. We can measure the brain activity and the heart, and we can see that this animal is no longer alive. But the complex thing is that the animal cells are still alive. Its heart has just stopped beating, and they will all die eventually, but the cells are still alive. So how do we get our head around that? Well, the thing is, we know cells. Cellular biology is a pretty old field, and cells have things in them that we can measure those things. So they have a nucleus, they have ER, they have plasma membrane, they have lysosomes, and we can measure the activity of those things. And if those things lose their activity, we can declare the cell dead. And that's what we call cytotoxicity assays or viability assays. And this is just some examples. Like, you can measure ATP, you can measure proliferation, You can measure LDH release or trypan blue increase. So we can measure those parts at that level, and then we can determine if the cell is dead or not. Now, we have to remember that the branch of toxicology is a little bit different than other biological branches, but I would say that toxicology really fits into the biological branch. But exposure, as I said before, is a really big part of it. So you need to be exposed to a compound before it can be toxic. So if there's no chance of exposure, there is no hazard. Or sorry, there's no chance of exposure, there's no risk involved. And you have to be exposed at a dose, at a concentration that causes a biological interaction. And that biological interaction has to be strong enough to cause a perturbation. And the perturbation has to also deplete the innate defense mechanisms of the cell, including things like glutathione and pumps and all that kind of stuff. And then you get a cellular perturbation. So you need to do a lot of damage before you get that far. And when you get that far, the cell is no longer unable to maintain normal cellular processes. So it will either go into necrosis, it will get a DNA mutation, or it will go into apoptosis. And these things we can measure very easy. And this is akin to morbidity and mortality in humans or disease. But what else happens is the cell is not evolutionary stupid. So it has adaptive responses that it's learned over 3 billion years of evolution, and it can react to this. So it can change its transcriptional programming based on what the perturbation was, and push it backwards to a normal biological state. Now, looking for us in culture, that doesn't happen fully. So the cells remain alive if we don't damage them too much, but they're in a stressed phenotype or a suboptimal function, and we term that as allostasis. So I wanted to talk a little bit about stress, because we call these adaptive response pathways are also called stress response pathways. And so stress in biology is a physiological disturbance or damage caused to an organism by adverse circumstances. And I actually forget where I was going to bring that. No, I'm not. I'm going to stop talking about that now. So yeah, these are all the things we have to consider: the exposure, the dose, the perturbation, the adverse effect, and measuring allostasis. And Barry, I'll leave it up to you now how long you want to give me.
Paul, keep going. Go ahead, please.
Okay, so I won't go into depth anymore on these slides, but these are all the targets, the cellular targets that you can hit, like DNA, lipid membrane, mitochondria, and they all have a specific consequence to the cell. Let's just stop with DNA. So a chemical can bind DNA, and that's not good if the chemical is binding with DNA, but it can also injure it by causing reactive oxygen species, and the DNA gets damaged, and the cell can try and fix it with the p53 pathway, but the effect anyway is aberrant DNA replication and DNA injury, and then this of course can cause mutations and can cause cancer. And kind of ironically, a lot of anti-cancer therapies also are carcinogens themselves, like cisplatin, because they kill the cell by binding DNA. And then there's a lot of biomarkers that we use in vivo, but we can also use them in vitro, and a lot of them transition from in vitro to in vivo. We're actually being able to develop biomarkers only from our in vitro experiments and backwards translate those to the clinic, which is really nice. Oh yeah, here's where I wanted to talk about the p53, the stress response pathways. So these are just some examples of adaptive stress response pathways that you might or might not have heard of. So p53, NRF2, the unfolded protein response, the metal stress response, HIF1 alpha. Then we have ones that are different. They're inflammatory stress responses. They're driven by NF-kappa B and STAT. And then we have ligand and xenobiotic activating pathways where the drug actually acts as the agonist. So the drug binds a receptor and turns it on, or is an antagonist to it. And not just a drug, sorry, it could be an environmental pollutant. And then we have lots of other kind of pathways that I won't go into. So these adaptive stress response pathways are tightly controlled. They have a specific transcription factor regulators, inhibitors, they have negative feedback loops, and they cross-interact with each other. And as I said, they're evolutionary ancient. So they're in nearly all plants, animals, and fungus, and they developed over billions of years of evolution. So I think I'm getting close to wrapping up. So if you think about toxicology, we have a human, a model human, and then we have the organs of the human, and we're interested in all of this. And then we have the cell, and then we have the cellular parts. And I've summed quite a lot of that up in a short review that's in Toxicology in Vitro, and the references there. I'm not going to go through these are, because I've already said it. I just want to quickly say about mitochondria, because mitochondria are actually, it's a weird part of evolution, that mitochondria were a kind of a prokaryotic organism that came into archaea about 3 billion years, no, it was less, I have it on the slide coming up. And so our cells have these basically other cells inside them. And this is a depiction of mitochondria from the MRC. And they're always moving and binding and dividing and fusing. And this is the tree of life. And you can see that where the mitochondria came in. So that was about 1.5 billion years ago. And I find it interesting that in toxicological research, we're now really going back to look at the mitochondria health and see how they are doing as well. So it's a nice, it's nice because it's a real part of biology. So this is just what a mitochondria is. So they're basically cell factories for producing ATP by eating up fatty acid and using oxygen to finally make the ADP into ATP. The mitochondria are different because they have their own DNA. So about 3% of the mitochondrial genes are their own, and you get this from the maternal genes. So from the oocyte has mitochondria, and you keep them all through your life because they keep dividing. And these are all the ways you can kill a mitochondria. So it's many ways to kill a mitochondria. And coming up to my last slides now. So we use all of this biological information, so genomics, transcriptomics, proteomics, metabolomics, to really understand what when we add a chemical to a cell, what's it doing? And the integration of these techniques is really helpful. We can't do it all the time, but every time we do an exercise, we learn a lot. And we're one of the most useful ones of those at the moment, because of the technology, is transcriptomics. Because transcriptomics tells you the intention of the cell. And the intention of the cell is what we're after. Because if you add a chemical and the cell intends to do a DNA stress response, it means it sensed DNA damage. So that's really useful. This is a publication, I think, if you have time and the capacity to read that, that was a wonderful paper we did together with Frederick Bois and Wolfgang Dekant. And there are so many names that I won't go through. But we integrated proteomics, metabolomics, and transcriptomics, and kinetics of the compound with these repeat tert cells that were grown for 14 days in repeat dose fashion. And it's a nice example of what I'm trying to say. So molecular toxicology today, yeah, basically it's what I said. So we take cells, we want the cells to be as good as possible, and then we add chemicals, and we look at metabolism and how the cell transports it, and then we investigate all the things I told you: nuclear receptors, signaling transduction, stress response pathways, mitochondrial toxicity, inflammatory processes, cytotoxicity, and omics and high contents. So we want to transition away from animals. Again, not really, because they're not useful. They are useful, but we think we can do better with human cells. And the time is really right now to do it now, because yeah, it's just everything seems to be in place, and I think we're good to go. So that's it, Barry. Thank you very much.